Hello, I am Cecilia Mir, and in this video I will talk to you about mutations. Okay, the main purpose is to identify and illustrate possible changes in nucleotide sequence in DNA and evaluate the significance of these changes in the resulting protein product. Does your body make errors? Yes, of course it does, and it's called mutations. A gene mutation is a permanent alteration in DNA sequence, as we will see here, that makes up a gene, such that the sequence differs from that is found in most people. This change occurs in our nucleotide sequence, as you can see in the figure. For example, this change, the original is GGT, and the change is GGC in the third nucleotide, for example. Or in the second, in another patient, for example, change from GGT to GTC. Okay, this change is due to mistakes when the DNA is copied or as a result of environmental factors such as radiations and mutagenic chemicals. The whole human family is one species with the same genes. Mutation creates slightly different versions of these same genes, called alleles. These small differences in DNA sequence, like hair color, like we will see, skin color, height, shape, behavior, and susceptibility to disease, make every individual unique. This way, genetic variation is useful because it helps population change over time. But not always mutation affects phenotype or physical appearances, as we will see. Variation mutation can also be inherited. Variation that help an organism survive and reproduce are passed on to the next generation. In the same way, variation that hinders survival and reproduction are eliminated from the population. This process of natural selection can lead to significant changes in the appearance, behavior, or physiology of individuals in a population in just a few generations, as observed here in the evolution of the peaks of the finches by Darwin. Here, the peaks. Okay? Once new alleles arise, meiosis and sexual reproduction combine different alleles in new ways to increase genetic variation. Okay. It's important to remember that losing the function of a gene doesn't always affect health. For example, most mammals have hundreds of genes that code for olfactory receptors, proteins that help us smell. Losing one of these genes probably doesn't make all that much difference. There are many more examples of variation that are neither good nor bad, but just different, like blood types, eye color, the shape of the lobe of the ear, as we see here. Some people have this type, another have the lobeless ear, or, for example, the presence of a dimple here, this beautiful girl, or what is called a widow's peak, as we can see in our friend Chris. Just like with diseased alleles, the process of mutation creates this more neutral variation. But with neutral variation, it can be impossible to tell which allele is the normal one that exists first and which is the mutant, and the distinction is often meaningless. It's useful to think of mutation as a process that creates genetic variation. We often refer to a mutation as a thing, the genetic variation itself. This approach can be useful when it comes to a gene associated with a disease. The disease allele carries a mutation, a DNA change that compromises the protein function. However, this approach gives mutation a bad name, and mutation is not always harmful. For example, the disorder sickle cell anemia is caused by a mutation in the gene that instructs the building of a protein called hemoglobin. As you all know, hemoglobin is the protein that carries oxygen through the blood. Okay, the normal adult hemoglobin has GAT in the original sequence, and for example, the mutant has GTG. This change in nucleotide causes a codon change in the RNA and a change of amino acid in the protein result. In the original hemoglobin protein, we have the amino acid glutamine here, glue, and in the mutated, we have valine. The change causes the red blood cells to become an abnormal, rigid, sickle shape, as we will see, the sickle shape. Okay, people with this mutation suffer from a disorder called sickle cell anemia, an inherited form of anemia. A condition in which there are enough healthy red blood cells to carry oxygen through your body. Okay. However, in African population, having this mutation also protects against malaria. So, mutation is not always harmful. As we have seen in the previous slide, mutation creates variation in protein coding portions of genes that can affect the protein itself, like here. But even more often, it creates variations in the switches that control when and where a protein is active and how much protein is made. That is the case that we observe here. Lactase. For example, lactase is an enzyme that helps infants break down lactose. Lactose is a sugar in milk built with galactose and glucose. Normally, the gene that codes for lactase is active in babies and they turn off at about age 4, more or less. When people who don't make lactase consume milk, they experience gas, nausea, discomfort due to the fermentation in the colon by the colonic bacteria. Because the lactose is not it's not good. There is an absorption for the intestine. But some people have a variation in a genetic switch that keeps the lactase gene active. So they can break milk lactose and absorb these products, galactose and glucose, without any problems. Another example is about the breed Shar-Pei. I love this kind of dogs. Thick, wrinkled skin defines the Shar-Pei dog breed. Puppies partially grow into their skin, but wrinkles remain on the adult head, neck, and back. Like all genes, one gene in special, HAS2, has switches that control when and where it's turned on or turned off. Here, an extra on switch makes the gene more active in skin cells. Sharpie makes too much this type of enzyme and therefore too much hyaluronan. The added hyaluronan thickens and skin and signal cells to divide more. The extra skin forms folds or wrinkles. We call this variation disease or both. This variation is in skin architecture. It's not a health problem, but the excess hyaluronan can cause an immune response, leading to skin inflammation and fever. However, mutation can also disrupt normal gene activity and cause disease like cancer. Cancer is the most common human genetic disease. It is caused by mutations occurring in a number of growth-controlling genes. Sometimes faulty cancer-causing genes can exist from birth, increasing a person's chance of getting cancer. Often cells can recognize any potential mutation-causing damage and repair it before it becomes a fixed mutation. DNA repair is a collection of processes by which a cell identifies and corrects damage to the DNA molecules that encode its genome. The rate of DNA repair is dependent on many factors, including the cell type, age of the cell, and extracellular environment. A cell that accumulates a large amount of DNA damage, or one that no longer effectively repairs this damage, can enter in one of three possible states, as we will see here. For example, first, senescence, an irreversible state of dormancy. Or, for example, another possible state is the cell suicide, also known as apoptosis or programmed cell death. And the last, cancer, unregulated cell division, which can lead to the formation of a tumor that is cancerous. The DNA repairability of a cell is built to the integrity of its genome and thus the normal functionality of that organism. Well, after this introduction, you can see the second part of this video where I will explain the causes, types, and effects of the mutation. Well, that's all now, but don't forget, once you stop learning, you start dying. Okay, thank you very much for your attention. See you soon. Bye.